Here we'll learn an overview of renal physiology. We'll focus on the structures involved in urine formation and the mechanisms of renal regulation. The kidneys are responsible for maintaining homeostatic body fluid composition, blood pressure, and a lot more. Indicate that they regulate the following: blood pH, blood volume, blood pressure via renin, and red blood cell formation. The kidneys are also responsible for detoxification of the blood and excretion of waste in the urine. Let's draw the structure of a nephron. It filters blood to produce urine. The nephron is often referred to as the functional unit of the kidney. There are 1 to 2 million nephrons in each kidney. First, draw a line and indicate that the renal cortex is above the line and the medulla is below it. Then outline the nephron. It begins as a ball-shaped structure in the cortex and continues as a wavy tubule that dips in and out of the medulla before emptying into a collecting duct. As we trace the flow of tubular fluid through the nephron, label its segments. First, show that an afferent vessel carries blood into the renal corpuscle. Here, water and solutes are filtered from the blood to create ultrafiltrate. Show that the efferent arteriole carries filtered blood away from the renal corpuscle. In the cortex, the efferent arteriole delivers blood to peritubular capillary beds. In the medulla, the nephron loop is accompanied by special capillaries called the vasa recta. Let's trace the path of urine formation through the nephron, starting with the ultrafiltrate in the renal corpuscle. Show that the ultrafiltrate exits the renal corpuscle as tubular fluid and enters the proximal convoluted tubule. From the proximal convoluted tubule, tubular fluid travels down, then back up the nephron loop. After exiting the loop, tubular fluid travels through the distal convoluted tubule and enters the collecting duct where it joins the tubular fluid from the other nephrons. Ultimately, the tubular fluid leaves the collecting duct as urine. So how does the tubular fluid become urine? As it travels through the nephron, it's fine-tuned via secretion and reabsorption of water and solutes, such as sodium and potassium, to form urine. Indicate that secretion is the addition of water and solutes from the blood to the filtrate. Reabsorption is the opposite. It's the removal of water and solutes from the filtrate. We discuss this in further detail in our notes. Ultimately, the urine created by the nephrons of the kidney collects and travels to the urinary bladder. Now let's take a closer look at the renal corpuscle, the site of ultrafiltrate formation. First, draw the glomerulus, which is the collection of capillaries that filter the blood to create ultrafiltrate. Then indicate that the afferent arteriole carries unfiltered blood to the glomerulus, and the efferent arteriole carries filtered blood away from the glomerulus. Show that the glomerulus is surrounded by the glomerular capsule. This capsule comprises a visceral layer which wraps tightly around the capillaries, and a parietal layer which creates the ball shape we drew earlier. Indicate that the capsular space is between the visceral and parietal layers. Ultrafiltrate collects in this space. It's continuous with the proximal tubule of the nephron. Now let's layer in some additional structures that help regulate blood flow through the glomerulus. If pressure were to drop too low, blood filtration would be inefficient. If it gets too high, it can damage the nephron itself. Indeed, high blood pressure is a major cause of kidney disease. First, show a nearby distal convoluted tubule and indicate the macula densa. This specialized area of the tubule senses when the salt concentration of tubular fluid gets too high. It's an indication of high blood flow through the glomerulus. In response, the macula densa releases chemicals that reduce the rate of blood flow through the glomerulus. Then show juxtaglomerular cells around the afferent arteriole. These cells sense when blood pressure through the afferent arteriole is too low and secrete a special enzyme called renin. It ultimately leads to increased blood pressure. Now let's shift to neural and hormonal control of blood flow through the nephron. Indicate that we'll learn 3 mechanisms that respond to low blood pressure and 1 that responds to high blood pressure. Start with ADH, antidiuretic hormone, which is released by the pituitary gland in the brain. Show that it increases water reabsorption by inserting special water channels called aquaporins in the nephron. As a result, the kidneys produce a smaller volume of urine. It's also more concentrated because the water has been selectively reabsorbed. Next, indicate that the renin-angiotensin system relies on the juxtaglomerular cells and the release of renin in response to low blood pressure. Show that renin converts a hormone called angiotensinogen to angiotensin 1. Angiotensin 1 is then converted to angiotensin 2 by another enzyme called ACE, angiotensin converting enzyme. Angiotensin 2 produces vasoconstriction and increases salt and water reabsorption in the kidney. This then raises blood pressure. Be aware that angiotensin 2 also triggers release of aldosterone, which promotes further sodium and water retention. As a clinical correlation, there are two key classes of blood pressure drugs, ACE inhibitors and angiotensin 2 receptor blockers. These drugs block this pathway and lower blood pressure. Next, show that the sympathetic nervous system releases norepinephrine in response to low blood pressure. Norepinephrine induces vasoconstriction and reduces blood flow through the kidneys. Lastly, show that in response to high blood pressure and stretching in the atria of the heart, ANP is released. ANP induces vasodilation and increases sodium and water secretion in the nephron. This lowers blood volume and pressure. Notice that ANP has the opposite effects of angiotensin 2. This concludes our diagram.